Slide 1. Peripheral circulation is a series of slide tape programs developed and produced by the American Physiological Society. This is Neural Control. Content expert, Dr. Paul C. Johnson. Please note that a printed study guide accompanies this program. It contains the full program text and references for additional study. 2. In this program, we discuss the sympathetic innervation of the vasculature, the synthesis and release of the sympathetic transmitter norepinephrine, the vascular smooth muscle response to sympathetic stimulation, the hemodynamic effects of nerve stimulation to an organ, and the effects of sympathetic activity on capillary exchange. 3. Before we begin this discussion, we should briefly review the topics covered in the earlier programs in this series that are important in understanding the concepts to be presented. In hemodynamics, we discussed the basic principles of fluid flow and examined the factors responsible for dissipation of pressure as expressed in Poiseuille's law. We then applied these principles to an analysis of blood flow, including the factors contributing to both viscous and turbulent resistance and the likelihood of turbulent flow as described by the Reynolds number. 4. In arterial and venous systems, we discussed the organization of the vascular network and the distribution of resistance throughout the network. 5. We also discussed the elastic properties of the vasculature, capacitance, and the force balance between transmural pressure and circumferential stress described by the Laplace law. 6. In exchange 1, the capillaries and solute diffusion, we described the anatomy of the terminal vessels of the microcirculation and the endothelial histology of capillaries, and we described the process of diffusion of solutes across the capillary wall. 7. In exchange 2, filtration and absorption of fluids, we described the manner in which fluid moves across the capillary walls, the forces that cause fluid movement, and the relationships between these forces and the membrane structure that determine the rate of fluid movement across capillary walls. 8. In Part 1 of Local Control, we discussed the functional and mechanical characteristics of vascular smooth muscle and described its response to distension. 9. In Part 2, we described the metabolic interactions between tissue and vasculature that balance blood flow and tissue function, and analyzed the combined effects of metabolic mechanisms and the myogenic response in 3 major phenomena: functional hyperemia, reactive hyperemia, and autoregulation. 10. Your first objective is to be able to describe the location and distribution of sympathetic innervation of the vasculature. 11. The autonomic nervous system controls the body's visceral functions. This includes the cardiovascular system and, in particular, the behavior of peripheral blood vessels. The system has 2 subdivisions. The sympathetic system controls the blood vessels in most organs of the body. The parasympathetic system is important in controlling the blood vessels of the external genitalia and salivary glands. 12. The sympathetic pathways emanate from the brain and pass down the spinal cord. At the thoracic and lumbar levels of the spinal cord, sympathetic fibers originate from cell bodies that lie within the lateral horns and distribute to the sympathetic chain adjacent to the cord. The sympathetic chain contains clusters of cell bodies or ganglia. 13. Preganglionic fibers from the spinal cord tracts form synaptic connections with cell bodies in these ganglia or with others in the periphery. The postganglionic fibers arising from the ganglia distribute to the blood vessels without further synaptic connections. 14. The sympathetic fibers involved in the control of blood vessels are found in the outer or adventitial layer of the vessel wall. Recall that the blood vessel wall consists of 3 layers: the adventitia, media, and intima. The nerve fibers run generally parallel to the layer of vascular smooth muscle. 15. The sympathetic nerve fibers have numerous swellings called varicosities along their length. The varicosities usually lie 0.1 to 2.0 microns from the smooth muscle cells. When the nerve is stimulated, the varicosities release the transmitter substance norepinephrine, which diffuses to the smooth muscle cells. 16. The density of innervation varies among different types of blood vessels. Large and small arteries are densely innervated. 17. Arterioles and precapillary sphincters are less densely innervated. Although nerves are found in the capillary region, their role is not clear. The capillaries themselves are not innervated. The innervation of venous vessels is generally sparser than that of the arterial network. Cutaneous veins, however, are a notable exception to this general rule. 18. Where do the sympathetic fibers that innervate the blood vessels arise? In what layer of the vessel wall are sympathetic nerves found? Do the sympathetic nerves make direct contact with vascular smooth muscle cells? Please stop the tape while you answer these questions and those that follow throughout the program. 19. The sympathetic fibers that innervate blood vessels arise in the sympathetic ganglia. Sympathetic nerves are found in the blood vessel adventitia. There is a separation of from 0.1 to 2.0 microns between the muscle cells and the varicosities, which contain the transmitter substance norepinephrine. 20. Your next objective is to be able to outline the synthesis, release, and fate of the sympathetic transmitter norepinephrine. 21. The nerve varicosities contain storage vesicles filled with norepinephrine, which is synthesized by the nerves. Norepinephrine is the transmitter that acts on vascular smooth muscle. When an electrical impulse travels down the nerve, the storage vesicles fuse with the cell membrane and release norepinephrine into the extracellular space. 22. Norepinephrine is produced within the nerve from tyrosine transported into the cell by an amine pump. In the neuroplasm, tyrosine is converted by tyrosine hydroxylase to dopa, and then by aromatic L-amino acid decarboxylase to dopamine. 23. Dopamine synthesized in the neuroplasm is brought into the storage vesicles by an active process. An enzyme present in the vesicle, dopamine beta hydroxylase, then converts dopamine into norepinephrine. 24. When an action potential depolarizes the nerve axon, calcium and sodium ions enter the neuron. Calcium is believed to cause movement of the storage vesicles to the cell membrane, where they fuse with the membrane and release their contents into the interstitial space by a process called exocytosis. The following slides describe the fate of the norepinephrine released from the storage vesicles. 25. It may diffuse into the interstitial space and bind to smooth muscle receptor sites, which are discussed in the next section, or it may be taken up by the vascular smooth muscle cell. The norepinephrine that is bound to receptor sites is released when interstitial concentrations fall. The norepinephrine that enters the smooth muscle cell is methylated and deaminated. 26. The norepinephrine may also diffuse into the surrounding tissue, where it is diluted below effective concentration or binds to collagen and elastin fibers. 27. A large fraction of the released norepinephrine diffuses across the blood vessel wall into the bloodstream. This route is especially important when the distance from the varicosity to the smooth muscle cell is large. Once it enters the blood, the norepinephrine is rapidly methylated and deaminated. 28. Finally, much of the norepinephrine released from the varicosity ultimately re-enters the nerve cell via the amine pump. This reuptake phenomenon is a principal mechanism by which norepinephrine is cleared from the interstitial space and is especially important when the separation between the varicosity and the smooth muscle cell is small. Pharmacologic agents that block reuptake prolong the action of the transmitter. 29. Describe schematically the process by which norepinephrine is synthesized. 30. Tyrosine is pumped into the nerve cell and converted to dopa and then to dopamine. Dopamine is transported into the storage vesicles where it is converted to norepinephrine by the enzyme dopamine beta hydroxylase. 31. Make a sketch of the process of norepinephrine release. 32. When the nerve cell is depolarized, calcium enters the cell, where it is believed to cause storage vesicles to migrate to the cell membrane. The vesicles fuse with the membrane and release the stored norepinephrine into the interstitial space by exocytosis. 33. What are the four fates of norepinephrine when it is released from the varicosities? 34. Norepinephrine released from varicosities may bind to or enter the smooth muscle cell. It may diffuse into surrounding tissue and bind to elastin and collagen. It may be carried away by the bloodstream, or it may re-enter the nerve cell. 35. Your next objective is to be able to describe the vascular smooth muscle response to sympathetic nerve stimulation. 36. When norepinephrine is released from the sympathetic nerves of blood vessels, it activates adrenergic receptors in the smooth muscle membrane. There are two major types of these receptors, alpha receptors and beta receptors. In vascular smooth muscle, the predominant action of norepinephrine is on the alpha receptors. 37. When alpha receptors are stimulated by the release of norepinephrine, they cause the vascular smooth muscle to contract, constricting the vessel. 38. Stimulation of beta receptors causes relaxation of vascular smooth muscle and vasodilation. This effect is normally overshadowed by the predominant effect of norepinephrine on the alpha receptors. When the alpha receptors are blocked pharmacologically, however, norepinephrine may cause vasodilation by beta receptor activation. 39. Norepinephrine can also affect smooth muscle indirectly. By changing the metabolism of the surrounding tissue, norepinephrine leads to the release of vasodilator metabolites. This is an important mechanism in the heart and possibly also in brain and liver. 40. We should mention that in some species, a special set of sympathetic fibers cause relaxation of arterioles in skeletal muscle. These fibers release acetylcholine, which is a vasodilator. This mechanism may be important in increasing blood flow to muscle before exercise begins. 41. Norepinephrine acts upon vascular smooth muscle through which of the following pathways? A. Excitation of alpha receptors. B. Excitation of beta receptors. C. Increases tissue metabolism. Which pathway is usually most important? 42. Norepinephrine acts upon vascular smooth muscle through all three pathways. Excitation of alpha receptors, choice A, is usually most important. 43. Your next objective is to be able to describe the contractile response of vascular smooth muscle following alpha receptor excitation. 44. Norepinephrine released from the nerve varicosities causes contraction by two means. First, norepinephrine acts on the cell membrane to increase permeability to calcium. Since calcium concentration is much higher outside the cell, a rapid influx of calcium occurs, triggering contraction. 45. Second, norepinephrine causes an increased release of calcium into the myoplasm from stores sequestered inside the cell. Activator calcium released from sarcoplasmic reticulum and the plasma membrane probably initiates contraction by binding to myosin. 46. Comparing the contraction response of vascular smooth muscle and striated muscle, we see that the initiation of smooth muscle response following nerve excitation is quite slow, several seconds, and the duration of the contraction is much longer. 47. The slow initiation of contraction in vascular smooth muscle is because norepinephrine must diffuse from the varicosity to the muscle cell, a distance of 0.1 to 2.0 microns, before it initiates the excitation-contraction process. 48. Vascular smooth muscle typically shows a two-stage response to norepinephrine. An initial contraction, probably due to intracellular calcium release, and a slower second contraction, which is probably due to influx of extracellular calcium into the cell. 49. In addition, norepinephrine levels in the neurovascular junction fall slowly. As described previously, reuptake into the nerve is one of the principal mechanisms for transmitter removal. In vascular smooth muscle, there is no mechanism for the rapid enzymatic destruction of the neurotransmitter, such as exists in the motor end plates of striated muscle. 50. What are the two pathways by which norepinephrine may cause arteriolar constriction? Why does vascular smooth muscle respond later than skeletal muscle to nerve stimulation? Why does smooth muscle contraction last longer than skeletal muscle contraction in response to a brief stimulus? 51. Norepinephrine causes arteriolar constriction by increasing cell membrane permeability to calcium and by causing the release of calcium from intracellular stores. Vascular smooth muscle responds to stimulation later than skeletal muscle because the norepinephrine must diffuse over a long distance. Vascular smooth muscle contraction lasts longer because some of the calcium that activates myosin diffuses from the extracellular space, and there is no enzyme to break down norepinephrine rapidly in the neurovascular junction. 52. Your next objective is to be able to describe the hemodynamic effects of sympathetic nerve stimulation to an organ and to compare the behavior of arteries and veins. 53. Sympathetic excitation of arterioles causes vasoconstriction, which is manifested by an increase in vascular resistance and a decrease in blood flow. 54. The magnitude of the vascular response is related to the intensity of stimulation. Here, the percent of the maximum attainable response is plotted against the stimulation frequency. Under resting conditions, there is a steady discharge over the sympathetic nerves at a rate of 1 to 3 impulses per second. This tonic resting activity produces an arteriolar resistance that is 20 to 40% of the maximum attainable response. Vascular resistance reaches its maximal value at a stimulus frequency of 12 to 16 impulses per second. This represents a 5 to 8-fold increase in resistance and a comparable decrease in flow. 55. Sympathetic nerve stimulation also causes venous constriction. This effect is manifested primarily as a decrease in organ blood volume. 56. Here, the percent venous response is plotted against stimulation frequency. Under resting conditions, the normal sympathetic discharge of 1 to 3 impulses per second produces 50 to 75% of the maximum attainable response. The decrease in organ blood volume reaches its maximum at a stimulus frequency of 8 impulses per second. 57. Comparing arteriolar and venous responses to sympathetic stimulation, you can see that the venous circuit is considerably closer to its maximally constricted state under resting sympathetic discharge, and that the arteriolar circuit has a somewhat greater capacity to respond when sympathetic activity increases. 58. Here we plot percent decrease of vessel diameter against the stimulus frequency. It is estimated that arteriolar vessels can constrict to a much greater degree than venous vessels. With maximal stimulation of 12 to 16 impulses per second, arterioles constrict by about 30%, while venous vessels constrict by about 15%. 59. If you cut the sympathetic nerves to an organ, would organ blood volume increase or decrease? Would blood flow increase or decrease? 60. If the sympathetic nerves to an organ were cut, organ blood volume would increase and blood flow would increase. 61. If you gradually increase the stimulation frequency, would the venous or arteriolar vessels reach their maximally constricted state first? At maximal stimulation, which vessels are more constricted? 62. When the frequency of stimulation is gradually increased, the venous vessels reach their maximally constricted state before arterioles. Venous vessels normally operate much closer to their maximal response level. With maximal stimulation, arteriolar vessels can constrict by about 30%, and the venous vessels by about 15%. 63. Your next objective is to be able to describe the effects of sympathetic activity on capillary exchange. 64. Although sympathetic nerves do not act directly on the capillary network, during sympathetic stimulation, capillary pressure usually decreases. This effect is secondary to the responses of the arterial and venous networks. Recall from the programs on exchange that capillary pressure is a major determinant in the filtration absorption process. 65. It is most important to recognize that capillary pressure is determined primarily by the ratio of venous resistance to arteriolar resistance. Since arteriolar resistance, RA, increases proportionately more than venous resistance, RV, during sympathetic stimulation, their ratio decreases. This would be reflected as a fall in capillary pressure. 66. Recall that when capillary blood flow falls, the plasma concentration and exchange rate of lipid-soluble substances, such as oxygen, falls. This is an example of flow-limited exchange, which may result from sympathetic stimulation. 67. Sympathetic stimulation may also stop flow in some arterioles, decreasing the number of flowing capillaries. This impairs exchange in two ways. First, it reduces the surface area available for exchange. 68. Second, a decrease in flowing capillaries increases the distance over which exchange of materials between blood and tissues must take place. Recall that this is critical for exchange of lipid-soluble substances such as oxygen and carbon dioxide. 69. Suppose that an organ is in an isovolumetric state. It is neither gaining nor losing volume because the Starling forces across the capillary are perfectly balanced. What would happen to interstitial fluid volume during sympathetic nerve stimulation? Why? 70. Sympathetic stimulation would cause the interstitial fluid volume to fall because the arterial vessels constrict more than the venous vessels, raising the precapillary to postcapillary resistance ratio, which lowers capillary pressure and favors fluid reabsorption. 71. How does sympathetic activity decrease capillary exchange of lipid-soluble substances? 72. By reducing flow in some capillaries, the exchange may become flow-limited. Sympathetic activity stops flow in some of the capillaries, which decreases the effective capillary surface area for exchange and increases the diffusion distance for exchange. 73. This concludes the presentation of neural control. We have discussed the synthesis and release of norepinephrine, the vascular smooth muscle response to sympathetic stimulation, the hemodynamic effects of nerve stimulation to an organ, and the effects of sympathetic activity on capillary exchange. To assess your understanding of the concepts that have been presented, and for a list of additional readings, please refer to your study guide. 74. This program is part of the Peripheral Circulation Series. It was developed and produced by the American Physiological Society. Copyright 1981.